We're including this in the video. That's my dog. Come on, hold the camera. No, like this. Itzel.
You said like this.
Get on it. No, your finger's in the video. You're not holding it right. The plushie would have done a better job. Yeah, okay. Ooh. Hey, your finger. What's so funny? This is my grade. Okay, so here we have... Can you stop laughing? So dog, she's... Hey, focus on me! Just hold it. Oh, you were holding it. No! Okay, so here we have a strand of DNA that is only found in prokaryotes, meaning bacterial cells or fungi. This, I'm about to present the Operon model function. So every cell contains all the DNA we need to produce all the proteins our body needs, but when cells become specialized cells, only certain cell genes will be turned on and off depending on what cell specialize in. But that's for eukaryotic cells, but here I'm presenting you prokaryotic cells, which only produce certain enzymes that are also proteins in order to aid in their function. Here I will be presenting a TRP operon. Regulation is important because it allows for certain proteins to be activated. An operon is the name of a few genes that are code for enzymes, also known as proteins. Here we have RNA polymerase, and here is a promoter. RNA polymerase is a builder and is needed to begin the process of transcription. It is the first step in protein synthesis. However, RNA polymerase needs guidance to know where to bind. This is where the promoter comes in handy, and the promoter is right here. So RNA polymerase would see this promoter and will latch onto it, and then will continue to go along the side and to make mRNA, which is the first step in protein synthesis, which is translation. Hold on, wait, what? Okay, once it's been binded, it allows for mRNA to be created, which then can start the second step, which is where was it transcription or translation? Detail, I don't know. Oh, it's translation. Yeah, it's translation, and that's where we create TRP, which is important especially in E. coli cells because it aids in the metabolism of the bacteria in order to break down sugars to use for energy. But when there is a repressor protein, and repressor proteins bind it with the operator, and it sort of halts the production of TRP because we don't always bacterial cells don't always need TRP present, and it's just wasteful to make something that a cell just doesn't need, and you're using up resources to make that protein instead, to make that protein when you don't need it. So when there's a receptor protein binded to the operator, the RNA polymerase can still bind to the promoter, but the repressor protein won't let it go past, and therefore it won't let it create mRNA that's needed to start protein synthesis, and therefore TRP won't be produced. So that's my model. Operons are typically activated. We can turn on and off. There are repressible operons, and there are also inducible operons. Repressible, like repress, to stop, or like, yeah, or inducible, which means to induce, to, start. This is my plushie. His name's Sake. He's a Niffler from Fantastic Beasts. I don't know if y'all watch it. Oh, Fantastic Beasts. It's a great movie. Great movie. It's my dog. Her tail. I need a vacuum.